is the cohort 7 Idea Accelerator Showcase. I can't believe there have been 7 cohorts already that are part of this Idea Accelerator. It's been wonderful. We have been so impressed and so excited by the work that the students have done that we, without a doubt, will continue to offer this Idea Accelerator going forward. Students are able to bring biology and engineering into their thinking and learn by putting their ideas into practice. And for the teachers too, I hope that it's been an exciting and interesting way to think about teaching biology, that is biology by building it.
The group decided to work on biomedicine. We are working on creating a treatment plan to help people with hepatitis B. We want to create a protein to help speed up the treatment and recovery process.
We just decided to focus on cleaning the pollution of water. The system we designed should clean the water and result in less body of polluted water around the world. The input could be the polluted water and the output could be a harmless cell releasing amyloids and opiates into the water.
The system we designed should be able to indicate a higher low risk of prostate cancer on the patient. The inputs could be the presence of CDC37, and the outputs could be changed in color, indicating whether or not a patient's at risk for or currently suffering from prostate cancer.
The system we designed should help produce more sugarcane with higher levels of sucrose. The input of the design is to amplify the process of photosynthesis, and the output of this design is to produce a larger amount of sucrose per sugarcane.
The system we designed should create less methane from the bovine by genetically modifying the cow's DNA to input methanotrose into its digestive system. The inputs are the methanotrose in the cow's gut, and the output is adding a genetic material that changes the color of the excrement.
We have a serious problem, and that problem is polystyrene pollution. Our solution is implementing a gene that can help worms degrade styrofoam in their digestive systems. For system levels, we're going to use the devices that are contained in DR11 and DR14. The strains also contain the parts we are using to degrade the styrofoam.
A problem, around 50 to 80% of crops were lost to pests globally from 2001 to 2003. This is our input, which is bacterium angioslo, which attacks the larvae of insects, to emit a blue glow indicating whether or not there was a pest infestation.
For our project, we decided to fight against the efforts of global warming by lessening the amount of methane production done by cows within their digestive system.
We decided to focus on a bacteria called Idionella sakaiusness, and it breaks down plastic. The only problem is it only really functions properly in controlled lab conditions. So we wanted to create an indicator that tells if it's still alive in certain environmental conditions. The input would be to detect life signs in the bacteria, and then the output would be to generate a mint smell indicating it's alive.
Our team decided to find a vaccine to slow and stop the spread of breast cancer. And we decided to design a system that slows the effects of breast cancer due to the abnormal growth of hormones.
We decided to focus on pancreatic cancer research because it is the fastest growing cancer on the planet. The system we designed will be able to destroy pancreatic cancer cells with the T cell we will create. Our input would be to create T cells to stop pancreatic cancer cells from growing, and the output would be to decrease the side effects of cancer cells or get rid of them completely.